Hello there and welcome to Smith and Sheridan on Biotech, a podcast on the science and business of biotechnology, presented by myself, Cormac Sheridan.
And me, Andy Smith. Hello, Cormac. Hello, everyone. How are you, Cormac?
I'm good, Andy. How are you?
I'm not too bad, thanks.
And today we're going to talk about a disease that is well known to people who look at the pharma and biotech industries, because it's been a very prominent disease for quite a few drug developers in the last decade and a half. But for people outside the industry, it's probably a disease they've never heard of unless they have a friend or a relative or a family member who is affected by this disease. It's called hereditary angioedema.
Can we do HAE?
We'll do HAE. And hereditary angioedema is kind of one of those largely genetic diseases that isn't as neatly defined as something like cystic fibrosis. Mutations in a couple of different genes can give rise to it, although primarily I presume it's the C1 esterase inhibitor is the main culprit. But there are others. I actually was reading that the gene encoding the blood coagulation factor 12 causes type 3 disease, and there's a few other diseases what's called type 3. And then there's types 1 and 2 are due to mutations in the SERPING1 gene that encodes the C1 esterase inhibitor. Do you want to talk me through the science, what's going on there at a molecular level that causes HAE, and what does it look like?
Well, yeah, and as you say, there are a number of genetic mutations that can either in the target gene or either upstream or even downstream, not just of the genes, but the genes encoding the inflammatory process. So, yeah, even forward in bradykinin, for example, or kallikrein, abnormalities in those can result in those. And then the patients then will suffer inflammation because the absence of this particular protein, this serine protease, can result in an event initiated by a trigger that results in inflammation. In many cases, 25% of cases, I think that trigger event is not known. It could be stress, it could be injury, but it results in inflammation in the body, which can be, people will know about anaphylactic reactions and the expression of the face that swells. But in HAE, it can be in the gastrointestinal tract. You can get blockages. But most frightening, it can be in the airways, the so-called laryngeal attacks that swell up so much that it blocks the airways and the patients can, unfortunately, asphyxiate. Otherwise, patients appear completely normal between attacks, either side of attacks when they subside. They're phenotypically normal. So if you're an HAE patient and you've been diagnosed, you've probably got that worry all the time about when's my next attack? How many attacks have I had this year? So it's probably just as much a worry as the actual disease is, when the next attack is, rather than having an attack.
And as with any disease, the variability is enormous, and some people have highly frequent attacks and others have very infrequent attacks, and the severity varies as well.
Which makes it difficult for companies developing interventions to address HAE, because obviously you want to recruit the most severe patients, but that's a very high bar, right? Patients with 2 attacks a month or 3 attacks a month or something, or 1 attack every 2 years, to demonstrate efficacy in those 2 different groups is going to be difficult.
And I know that you were prompted to discuss this one this week because of a very interesting new paper that came out in New England Journal of Medicine from Intellia Therapeutics, suggesting that their gene editing approach could have profound efficacy in patients.
Yeah, and it's early days though.
Early days. And before we get onto all of that, we really should look at the sort of history of drug development here for the last 15 years or so, because you could really strongly make the point that HAE is one of the most successful examples of the orphan drug designation really working for patients, because it is a rare disease. I think the incidence is about 1 in 50,000 or something.
Yeah. And
there's an awful lot of therapies have been developed in the last 15 years or so. There's been a bunch of plasma-derived C1 esterase inhibitors that are basically the human protein isolated from pooled plasma taken from multiple donors. And that's a very traditional type of a way of getting a biologic drug on the market. But there's been a few there. Cinryze from Lev Pharmaceuticals, acquired by Shire, acquired by Takeda.
With Vira Pharma in the middle, yeah.
And which one in the middle?
Vira Pharma bought Lev, and then Shire bought one.
Thank you for that piece of history.
Bit incestuous, isn't it? But yeah.
And then there's Berinert from CSL Behring. They have a plasma product. They've updated it with Haegarda, which is a sub-Q or subcutaneously delivered plasma-derived product. And then you also had, and this is the one that was my entry, if you like, to the HAE story. I've been following the Dutch company Farming for quite a while.
Mm.
And of course, famously, they were the second to market, not the first, with a biologic drug derived from the milk of a transgenic animal. They've genetically engineered rabbits to produce a recombinant C1 esterase inhibitor in their milk. And you milk the rabbits and you get your drug while you purify it from the milk and you don't drink it. And it took them a very long time to get that drug approved. And they were initially going to be the first to market with a recombinant product, and they kept on losing ground to other competitors. But they got there eventually, and they do have a successful product franchise in their hand. And then there's a whole bunch of other ones. There's Firazyr, aka Acatabant, the German company Jerini, acquired by Shire, acquired by Takeda. That's it's A begat B begat C. Old Testament stuff, really. That's a bradykinin B2 receptor antagonist. So that prevents bradykinin, which is the key culprit that triggers these attacks at the blood vessel level. It's a vasodilator, and it causes the blood vessels to leak out, and excessive fluid accumulates. You get edema, swelling, inflammation, pain.
So that's downstream of C1 esterase, right?
Yeah, exactly. Yeah, C1 esterase is responsible for, let me see. What's C1 esterase responsible for?
All the inflammation above, but upstream of bradykinin B2 then.
You also have kallikrein, and that's involved. So, and I don't know the relationship between C1 esterase and kallikrein, actually.
I think kallikrein is downstream of C1 esterase. I don't know whether it comes before bradykinin or...
Bradykinin is the final, that's the molecule, that's the culprit.
Right, so it could be that kallikrein comes in between the two. But in any event, you've got an inflammation cascade cartoon, have you?
Yeah, and it's a very complicated one.
Well, I mean, the thing is, there's probably crosstalk between the two. So if you inhibit one step, that doesn't mean that you eliminate all inflammation. On the other hand, I suppose you've got patients who've got, I think it's bradykinin deficiency, and they're again phenotypically normal. They don't have clotting.
Yeah, I know that. That's why
kallikrein deficiency. Yeah.
Yeah, so it turns out that the disease primarily results from a deficiency or a lack of functional C1 esterase inhibitor. And that deficiency, it leads to a dysregulation of a cascade that ultimately leads to the formation of bradykinin, which is a vasodilatory signal molecule.
Right.
So when you have excess bradykinin sloshing around in your system, triggered by the cascade, as you were saying, the triggers are not necessarily fully identified or understood, but when it does happen, you basically get this leakage of the blood vessels, you get edema, swelling, fluid release, inflammation, pain. And so there's various points of intervention. Kallikrein is an important intermediate protein involved in the whole process, and that too has been targeted by various drug developers. You have both antibody-based and small molecule oral-based drugs, and there's some new genetic gene-based medicines also targeting production of either pre-kallikrein, which is the inactive form, or kallikrein, the active form. And basically, when you knock out kallikrein, that cascade, you arrest bradykinin production because it's basically further up the pathway again. So there's been a whole string of drugs. Jerini from Germany was a company that sort of pioneered the whole mechanism of bradykinin receptor antagonism, which I guess is sort of at the final hurdle, as it were.
And I was an investor in Jerini as a private German company when I was at 3i. And like you, before that stage, I'd never really heard of HAE. I mean, I'm ashamed to say I invested in CSL, Australian plasma-derived manufacturer. But the treatment of HAE, its product for treatment of HAE, was never really part of the investment thesis. It was tiny product. And I saw Lev Pharmaceuticals before Shire bought them, but 3i gave me exposure to Jerini, which is a smaller molecule. And it has some advantages we might get around to talking about in terms of these, because if you're going to have a product, which Jerini's product was for attacks, treatment rather than prevention, then you want it to be easily available, right? So Jerini's product was a self-injected, room temperature stable product, Whereas all those plasma-derived products, like even FP frozen plasma, they have to be stored at 4 degrees, not frozen. FFP is frozen. Some of them have to be stored at 4 degrees, and you can't sort of carry them around or keep them in the medicine cabinet at home. So that's a distinction, or at least a segmentation, if you like, from products that were used and are still used in the prevention of attacks. And obviously that's a panacea, right? Because you want something that lasts a long time and gives overall coverage, so you don't fear going out and be exposed to something that might result in an attack. Whereas if you have a product that's only for treatment, then the patients have to carry it with them. It's on demand. Yeah. And the actual prevention market is a lot bigger, obviously, because if you can be effective preventative, you've got a guaranteed market of patients there. Whereas the number of attacks, as you said, varies amongst patients. So that's a differing market.
And an interesting kind of real world insight into what's been used for prevention, because I mean, most of the drugs on the market, they originally were approved for prevention of acute attacks. And then they eventually maybe got a line extension for prevention. But whether they're actually used for prevention or not is a moot point. And I think you raise very valid points that they don't lend themselves well to they do lend themselves well to the prevention thing if you just take it at the required dose.
Yeah, but they won't have the serum half-life of the natural products that are plasma derived, that are naturally circulating in most of us. Yeah. And even the Jerini Shire Takeda product, it's a small molecule peptide, so it's going to have a short half-life. So that's why it's for attacks rather than for prevention.
Well, it was interesting. I was looking at the New England Journal of Medicine paper, which Intelia and its clinical collaborators published end of January there. And just as a by the by, we'll get onto that paper and the data later, but the patients and it's only 10 patients that we're talking about, but they were allowed concomitant use of prophylactic therapy. And of the 10 patients that are being discussed in the paper, 6 of them were on concomitant use of long-term prophylaxis. And now I don't have the numbers in front of me, but I can check. But in most cases, they were either on a steroid or a relatively recently approved small molecule drug from Biotech, or Deleo, or berotralstat, which is an oral plasma kallikrein inhibitor. So that's an oral drug, and you take it at whatever the dose interval is. I haven't checked, but I presume it's once daily or something like that. So that's interesting to me, just to see what people were actually using for prophylaxis. But what's interesting to me, though, is that the drugs, and I mean, Takeda as well had a promising antibody-based product called Tacrizo, or lanadelumab, an antibody-based plasma kallikrein inhibitor approved back in August 2018. That was going to be regarded as being quite competitive. Biotech obviously is in there too. So the market is well served. These drugs are effective. They're not cures, but they do reduce the frequency of attacks considerably. And CSL Behring is still in play, and they have an antibody that also looks kind of interesting. It's under review at both the FDA and the EMA at the moment. It's called garadacimab, and that targets activated factor 12. So it goes to the very top of the cascade that results downstream in the overproduction of bradykinin. So they're still in play, and I had a quick look at that, and that's high levels of efficacy, more or less 0 attacks in patients on the drug for the reported period. So at the same time, there's been a whole lot of new entrants with gene-based approaches into the space. And not only is it Intelia with a CRISPR-Cas9 based editing approach, but Astria Therapeutics they're actually a long-acting antibody, so they're still in clinical trials. But Adarex Pharmaceuticals with an siRNA, they're targeting the pre-kallikrein, the inactive form of kallikrein. And Ionis is also in there, and they also have a product which is in phase 3, an antisense nucleotide also targeting pre-kallikrein or PKK. That's called donidalorsen. It takes a while to get your mouth around these products. So Intelia has all the sexy CRISPR thing going on, and it has all the excitement, but also some of the question marks in terms of safety. I mean, just to go through it though, for those who haven't seen the news, of the 10 patients treated, they were across 3 dose groups. They received either 25 milligrams, 50 milligrams, or 75 milligrams of the ribonucleoprotein complex, and they're editing the KLKB1 locus, which basically encodes kallikrein. So they're basically trying to knock out that protein. And apparently they state in the paper that there are people walking around who are knockouts for that protein and they're perfectly healthy. They have no kind of unusual features in terms of their coagulation.
And if you take their blood to the lab and run some clotting factors, you can't tell they're not normal.
Yeah, so that's interesting.
But we've talked about all these drugs, many of them plasma derived, most of them expensive. And then the Intellia's publication, Intellia's presentations, and for full disclosure purposes, I do hold some Intellia shares in one of my pensions somewhere.
Okay.
That must have sent shockwaves through all these companies that are developing them, because, I mean, I just think they're not cheap plasma-derived products. You need humans or rabbits or recombinant cells to produce them. And then patients need to take them if they're prophylactic for the rest of their life. That's going to be quite a cost. And you can see companies like Intellia and other gene therapy companies rubbing their hands thinking, just think how much we could cost this one and done therapy to effectively provide prevention. The rub of that is that it doesn't appear to be one and done. If it is one and done, in the 10 patients we've seen so far, it prevented 95% of the attacks. Yeah. And this is an interesting concept that I'd like us to spend a few minutes on, is that, people who invest in gene therapy companies think these are great things. You can have a one and done, one shot therapy that saves a lifetime of probably very expensive drugs and caring costs and things. And that's the original promise of gene therapy. Yeah. In this particular case, the LNP,
the lipid nanoparticle.
Yeah, as we talked about before, makes its way to the liver and then does its work, edits the genes in liver cells, but probably doesn't do it in all liver cells, right? No. So that's why I suppose you don't get complete coverage. On the other hand, you look at things like the early studies on gene therapy of haemophilia, and even patients who receive recombinant factors, they don't need 100% of what a normal person's clotting factor is. And we've seen the converse is also true. So those patients mentioned in that Intellia paper with a knockout in pre-kallikrein, they're phenotypically normal and they don't have clotting either. So perhaps the one and done aspect of gene editing and gene therapy can still be efficacious, even if it doesn't give 100% of the normal whatever factor it's in.
Yeah. I think the primary endpoint on the paper was the mean reduction in kallikrein protein levels. And there's a very nice dose response curve. The low dose arm, there was a 67% reduction. In the middle dose range, it was 84%. And the high dose arm, it was 95%. Mm-hmm. But there wasn't a similar dose response in attacks. Terms of the clinical attacks, because they've also stated, and I think it's already stated in the literature, that if you get a reduction in kallikrein levels of about 60%, that threshold is regarded as being protective.
Yep. You're done anyway. But then can you imagine the clinical operations people seeing those results and seeing that the up and down dose response, thinking, oh no, how are we going to make something out of this?
Yeah, I think though it shows that it's always, I think, very reassuring for people who produce a study that it is in agreement with prior art, as it were. And they are obviously pushing the boundaries in terms of the potency of the effect. Because what I think is extraordinarily important about this study is that they were getting these changes of about 95% pooled across the three dose arms. That was in the case of 60% of the patients who were already on prophylactic therapy. This is not a placebo effect, even though it's not a placebo-controlled trial.
There was no placebo arm, right? Yeah.
There's no placebo arm. But the point I'm making, though, is that you could argue that it's almost like head-to-head versus prophylaxis, because they're getting additional benefit on top of prophylaxis.
Yeah, but then they come off the prophylaxis, didn't they, in part of the follow-up study?
They did, and had no further attacks. Now,
so on the one hand, people could criticize this study. A, it's only 10 patients, right? B, you look at the historical attack rate, and we all have come across in oncology studies historical controls that don't work because the standard of care changes over time. So whatever your rate of mortality was in the past with these particular type of cancer patients, it's not today, because oncologists are better at treating cancer than they've been before. So there's that aspect to it. But the fact that these 10 patients are their own controls, so they have an attack rate before, which is historical and patient reported, they have the gene therapy, and then investigators measure it over a period, and that attack rate is lowered, is not quite the placebo-controlled panacea, but it's getting there, isn't it?
It's good. I mean, just for the sake of argument, the median number of attacks during the historical attack period were 7 in the low dose arm, 3 in the middle dose arm, and 14 in the high dose arm. So these are people with active disease,
Yeah.
and you're getting, roughly speaking, a 95. I mean, the best response was 98% in the middle dose arm. So 95, 98%, that's profound efficacy.
And it raises another interesting issue as far as gene editing and gene therapy of HAE is concerned, is that, these patients, these 10 patients in this phase 1/2 study, they are probably the patients, if you're an allergist, you worry about the most, right? Because they have so many attacks.
Yes.
But as you said in your introduction, there's a whole spectrum of mutations that result in patients who have had one attack every 2 years or one attack in 5 years. Are they patients for gene editing? Or are they patients for an on-demand therapy? Or are they patients for recombinant plasma-derived preventative therapies? I'm guessing that the fewer attacks you have, the less likely you are going to be interested in, and your insurance company or your payer is less likely to be interested in paying a lifetime therapy to prevent one attack every 5 years. It's a bit like statisticians, medical statisticians have this number needed to treat, or in vaccinations, number needed to vaccinate in order to prevent the disease. We're going to have that sort of calculation go on in payers' minds if these gene editing therapies ever get to the market for HAE.
Yeah. But what's fantastic about it, though, because it's a lipid nanoparticle that goes to the liver, this is an in vivo therapy. So it's inherently scalable. It's, in theory at least, as easy as giving somebody an mRNA-based vaccine in a nanoparticle. Now, because it's gene editing therapy, there will have to be considerable follow-up just to make sure that there aren't any untoward effects arising from off-target edits leading to potential nasties down the line. There's at least a theoretical concern that if you introduce a double strand break in a DNA molecule, that you might get some sort of chromosomal rearrangement that could result into some sort of malignancy.
That's very good, Cormac. You're covering the aspect of holistic safety that very few people worry about when they look at companies. And
with CRISPR-Cas9, you have to.
Actually, and with all gene therapies, the FDA mandates at this point in time a 15-year follow-up of those patients who received that in order to look for aberrant mutations or insertional events that happen when you have this sort of therapy.
Yes, exactly. And I mean, and I hasten to add, it's a theoretical concern. It's not something that's necessary.
It is, but as you've also hinted at, LNPs are the vehicles for the Pfizer and the Moderna mRNA vaccines. They have a safety database now of billions of people. And the paper actually says that the only side effects they see were injection associated with the LNPs rather than the CRISPR-Cas9.
Precisely. So far, it looks rather good. And yet, there are other approaches. Old friends Ionis Pharmaceuticals are hoisting the antisense oligonucleotide flag in this space as well. And also, as we mentioned earlier, there's siRNA program as well. That's just started the clinic last month, in fact, from ADRX Pharmaceuticals. So these approaches are neat because if you have an active molecule, you don't have the concerns about long-term theoretical safety implications, but you do have the same level of precision that you have with gene editing, that you're knocking out PKK. So you're just doing it at a different part of the production process than is the case with the gene editing approach. So it's a very potentially compelling offering as well. And we have to see data from the ADRX program, but the Ionis program, that's a very, very active agent. I think that they've, in a phase 2 at least, they had a 90% reduction in HAE attacks per month in actively treated patients versus placebo. That was after one dose. After 2 doses, it was a 97% reduction in monthly attacks and a 96% reduction in moderate to severe attacks. And all but one of the patients in this 20-patient cohort were attack-free, 92.3%. So that's kind of Intelia-like efficacy.
It is. I mean, it appears amongst a lot of things in modern medicine and bioscience. There are no absolutes here, right?
Yeah,
I mean,
yeah,
in fact, I think in that New England Journal of Medicine patient, you only need a target of 60% knockdown in kallikrein in
yeah,
order to be efficacious. Now, whether
yeah.
you achieve that through a one-off gene editing intervention
Hmm.
or the other end of the spectrum, plasma derived proteins or long acting siRNA or long acting antisense molecules, depends on the number of attacks the patients have every year. So there is space for everyone, I believe. But I think in gene editing, with that one-off aspect and that already high efficacy rate, it's going to be complicated by whatever price Intellia charge for that one-off product.
Yeah,
If it's going to be $10,000, which it won't be, but if it's
no,
going to be $10,000, then you're going to think payers are going to love that because
yeah,
I mean that's probably an annual cost of a recombinant or a plasma-derived therapy. But if it's $50,000 or $200,000, if you've only had one or two attacks in the last 3 or 4 years, then payers are going to say, and their physicians are going to say, well, this long-acting siRNA that has a similar efficacy, and it won't cost us that much over that period. So it's going to come, oh no, Cormac, it's going to come down to money again, isn't it?
yeah. And I mean, the money is a lot of money.
Yeah, yeah, yeah.
Just a quick and dirty search. I mean, Haegarda, which is the sub-Q, so the modern plasma-derived product from CSL Behring, it's about $500,000 in the US.
But everything's $500,000 in the US.
Yeah.
What's the European price or the UK price? Yeah.
This is serious cash. I mean, I presume the cost of goods for the Intellia product is actually a whole lot cheaper than the cost of goods for a plasma-derived product, which is a very cumbersome production process.
Oh, it's de minimis, I think. I mean, it's not de minimis, right? Because you have to produce it sterile, you have to produce it in a form that is storage and quality controlled and things. But yeah, it's not $500,000, right? It's not 30 years at $15 million. It's not a lifetime therapy. It's a fraction of that. Now, Intellia would argue, what about the costs of these clinical studies to get those patients with that high number of attacks in to pay for their preventative therapies while we wean them off or while the gene therapy has an effect? Is not cheap either. So the costs of running the clinical studies for a number of years will be much more than the cost of the drug. The price of the drug, on the other hand, will then reflect that and what comes after it.
Yeah, I mean, none of them are cheap. I mean, that we can take that as read. Ordeleo costs more than $300,000. It's about $310,000.
Yeah.
So these are US prices. But
so you can see why this is a fruitful area to biotech companies, because they see small number of patients, easy to find because they're cared for by a small number of physicians, a product that is a one and done therapy that effectively saves a lifetime of very expensive therapies. That's something that should appeal. If you're a biotech company and that doesn't appeal to you, you're in the wrong business.
yeah, and what we can say, I mean, I will never stand over the cost of pharmaceuticals. I always believe that they could be cheaper and people will still make money. Leaving all that aside, that's a whole other day's argument or debate. But you really could argue, though, that HAE is one area where biotech innovation is working.
Yeah. I mean, even from the earliest days of the plasma-derived stuff to icatibant to last week's publication in New England Journal of Medicine.
It's all in there. We've got
Yeah,
antisense,
yeah.
we've got antibodies, orals, plasma-derived products. We've got one of the few transgenic animals. It's almost like a sort of a mini history of the entire biotech industry.
Yeah, yeah. We haven't mentioned there is some survivorship bias there, Cormac. So there must have been products that have been tried and didn't get anywhere. But let's
Yes.
ignore those for the moment.
Yeah, we don't have enough time in the world to discuss those as well. But to the survivors, and most of all to the patients, I say 3 cheers for biotech innovation. When it works quite nicely.
Yeah, yeah, absolutely.
Yeah. Okay. Until the next time.
Bye-bye, everyone. Bye-bye, Cormac.